Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen, distinguished officials of the Ministry of Health, respected moderators and invited speakers. In this beautiful afternoon, we would like to welcome you all to attend this webinar hosted by the National Institute of Health Research and Development, Ministry of Health, Republic of Indonesia. For approximately 2 hours, we will gain insight, knowledge sharing, and evidences and experiences, particularly in describing the laboratory collaborations and clinical trial of vaccine and antivirals in Indonesia to control the COVID-19 pandemic. In this webinar, I would like to gently remind the audiences to value respect and courtesy at the most during our webinar sessions. And the link of the evaluations form will be given at 15 minutes before the end of this webinar. So attendances, please stay tuned for it, as we will use the informations in the attendance form filled in that form as for e-certificate purposes. Without further ado, I would like to invite the Director of Center for Research and Development of Biomedical and Basic Health Technology, Dr. Vivi Setiawaty, Master of Science, to deliver welcome remarks and officially open this webinar. Ibu Vivi will give the speech on the behalf of the Acting Director of the NIHRD. The distinguished Ibu Vivi Setiawaty I would like to invite you to talk, and please, the time is yours.
Assalamualaikum. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, honorable speakers, moderator, and participants. Allow me on behalf of National Institute of Health Research and Development, Ministry of Health, Republic of Indonesia, to greet you with a warm welcome to the third international webinar, focusing on ending the pandemic COVID-19, efforts and challenges. I would also like to express my gratitude for the presence of the moderator, speakers, and participants in this webinar. COVID-19 has been announced as public health emergency of international concern on 30 January 2020 by WHO. Later, on 11 March 2020, after conducting assessment, WHO stated that COVID-19 can be characterized as a pandemic. Globally, as the 25th August 2020, there have been 23 million confirmed cases with 800 deaths reported to WHO. Meanwhile, since its first confirmed case in the beginning of March 2020, Indonesia has 150,000 confirmed COVID-19 cases with 606,000 deaths as 25th August 2020. Indeed, COVID-19 has become a national as well as global issue. Honorable participants, public health intervention is considered as the key to mitigate the trend of increasing COVID-19 cases and hopefully in the future to end the COVID-19 pandemic. The implementation of public health intervention includes active case finding, contact tracing, optimal health care delivery in all levels of healthcare facilities, conducting infection prevention and also control measures, and developing vaccine and drugs, along with other alternative therapeutic methods should be implemented simultaneously. The National Institute of Health Research and Development, as one of the institute under Ministry of Health Republic of Indonesia, has been actively implementing these actions by working together with related counterparts, including those within the laboratory network in Indonesia. Other part of public health intervention that equally essential to encounter COVID-19 issues is the research and development of COVID-19 vaccine. According to the draft landscape of COVID-19 candidate vaccines published by WHO in August 10, 2020, there have been 28 vaccine candidates tested in clinical trials. 6 of them is now entering phase 3. The vaccine platform developed in many countries varies from non-replicating viral vector inactivated RNA, protein subunit, and DNA. Indonesia's government, research institute, universities, and pharmaceutical companies also participate in some of these vaccine candidate trials. Honorable speakers, moderator, and respected participants, I sincerely hope this online seminar, we can learn and gain more insight about laboratory collaboration and vaccine candidates clinical trials in Indonesia as well as in other countries. Specifically, through this webinar, we will be able to share knowledge of COVID-19 biomolecular testing update and COVID-19 vaccine candidates clinical trial progress conducting in Indonesia. This is to share information on global experiences and also laboratory networking to get better understanding of challenges Indonesia has to face in developing COVID-19 laboratory network. I hope the discussion will be fruitful as well as and together we can overcome the COVID-19 pandemic. Thank you and wassalamualaikum warahmatullahi wabarakatuh. By this end of the speech, I officially open this webinar, international webinar of COVID-19. Thank you.
Thank you, Bu Vivi, for your kind welcome. Now it's time for us to start the main sessions of this webinar. Respected audiences, ladies and gentlemen, there is something special in this webinar. First, for attendances in the Zoom webinar, we have provided a translator to translate Bahasa Indonesia to English and vice versa. And Zoom attendants can click the interpretations facility on your screen to listen to the translator. But to all attendants in YouTube channel, you will listen to the original language delivered by the speakers. And this webinar will be divided in two sessions. The first three speakers will be moderated by Prof. Tjandra Yoga Aditama. He is a senior advisor and an acting director of communicable diseases to the World Health Organizations of the Southeast Asia Regional Office, a professor in pulmonology and respiratory medicine, Faculty of Medicine, University of Indonesia, and also an adjunct professor in the Center for Environment and Populations Health, Griffith University, Australia. So, ladies and gentlemen, allow me to invite Prof. Tjandra Yoga Aditama and pass the first sessions to him. Prof. Tjandra, are you already tuned into our sessions? And I would like to deliver this session to you.
Thank you, Bu, because my mic and my video was mute, but now it's open. So thank you very much. Thank you very much. So, colleagues, ladies and gentlemen, we will begin with the first session of this international webinar, and we will have three presentation during this first session, as already mentioned. The first one will be from Prof. Nyoman from Airlangga University. The second one will be from Ibu Vivi, the director from NIHRD, who just delivered the opening speech. And the third one will be from Dr. Bahl from my office in WHO SEARO. Each of the presenters will make the presentation about 20 minutes. And at the end of the 3 times 20 minutes, so at the end of 1 hour, we will have another 20 minutes for discussion. So let's begin with the first presentation. So the first presentation will be from Professor Nyoman Puspaningsih. The organizer, please kindly upload her CV. So as you can see here, Prof. Nyoman is now is a biochemistry lecturer at the Department of Chemistry, Faculty of Science and Technology, Universitas Airlangga. And she already published research, also awarded 2 national patents. So, and she has a quite extensive educational background and then experience. So, Professor Nyoman will speak about the globally biomolecular of COVID-19. Professor Nyoman, over to you, please.
Thank you, Professor Tjandra. Assalamualaikum warahmatullahi wabarakatuh. The Honorable Dr. Vivi Setiawaty, the Head of Center of Biomedical and Basic Health Technology and NIHRD Indonesia, and also the Honorable the keynote speaker in this today, the international webinar. So thank you very much for all organizing committee to invite Universitas Airlangga on behalf of Universitas Airlangga, I will also share my presentation and also our research in Universitas Airlangga in this kind international webinar ending the pandemic COVID-19 effort and also challenges. So please allow me to share my screen, Professor Tjandra. So I hope that this is already seen by all the participants.
Yes.
Sorry. Because we have a very limited time, so I would like directly to continue my presentation, and then the title is Globally Biomolecular COVID-19. So I think all of the participants who joined this international webinar already known and also already familiar with this kind of the SARS-CoV-2 genome, the new one of the COVID-19. So we know that all the viruses already have the compartment of this this kind of the virus. Also with the COVID-2 genome, that we know that this kind of the SARS-CoV-2 or COVID-19 is have a more bigger size, is around 30 kilobase in size, and also have 12 functional operating frame. So this kind of virus is the single strand positive RNA. So then why is very important to us to study about more detail of this kind of the virus. So when we look for this kind of the map of this genome virus of the COVID-19, we can see that this location is very important to produce two kind of the protein. This is main protease and also the RNA polymerase. This is will become the target of the drug. And then the other side is this gene is encoding the spike and envelope, the membrane, and also nucleo protein of this virus with this kind of the structure. So if we look more deeply in the information of the genome, whole genome sequence of this virus, especially for the worldwide, we can see here now that in GSAID, the database of the global COVID-2 genome sequence information, we can see here that now they have already collected around 84,000 viruses in this kind of the data base. So how about Indonesia? We are still very limited information that we submitted to the kind of the GSAID database data for Indonesian virus information. So we can see until today, I just checked also in GSAID, that now we have around 33 whole genome sequence information submitted in the GSAID. By this kind of the collection, we have only 21 that whole genome sequence already finished. But the other is only partially sequencing by many kind of institution in Indonesia. Also, it included Universitas Airlangga. So when we see Universitas Airlangga have already submitted 6 whole genome sequences of this kind of the virus, COVID-19, and you can look here, the ID of our university submitted data is EG is Java, and ITD is our Institute for Tropical Disease in Universitas Airlangga. So then we already have submitted 6 kind of the whole genome virus. So what is the important or interesting for us to study more detail about this kind of the COVID-19? So especially for the globally biomolecular aspect. So if we then see in this kind of the information, we are very, very interesting to study more detail about the mutation, the point mutation that happen in this kind of the virus. So then I think in this today, so many issues then appear regarding to the point mutation of the in the spike location from D to G, that we can see also here the position of the mutation. So I think for the mutation, I think all participants, I hope that agree with me that we are very interested in frame mutation. So it's mean that can be very changing the virus properties or biochemical or biochemistry or biological or medical information regarding to the in-frame mutation, because the in-frame it's mean that there will be changes the kind of the frame of the genomic information of the virus can be to deletion or insertion. So because we know that the codon information will be partly code of the amino acid. So it's mean when it will be happen for deletion or insertion of the gene, so it will be changing the amino acid for maybe for this kind of the matter. So it can be also changing the property of the virus, the infectivity, or maybe also the pathogenities of this virus. So this is we are very really interesting in this kind of the information. So when we look here, so we know that the vaccine is a target is majority in the spike, but sometimes also many people also designing by the epitope or any other platform. But if we look here, that we can see the mutation location is almost for the all important protein of the virus, like spike, envelope, membrane, and also nuclear protein. And then this kind of the mutation is also already informed in the GSET database. So from the many kind of the mutation, then we look more deeply in the kind of the changes for the wild type to the mutation of the D to the G. So this position is in the 614, and we can see this is started this kind of the mutation in China in January 2020. So what happened now? So now this kind of this point mutation is becoming very massive. So we can see here that 77% is regarding to point mutation of the D to the G. So almost 63,000 infected patients have this virus mutant or variant. So this is also refers to the in the location of the spike protein. Information from the GSET 2020. Recently, this information is very different with the when we check last April. This is still in 30,000, but now it's becoming double, around 63,000 times, and 77% of for the whole 84,000 collecting virus genome sequence in GSET. 77% is mutated happen in the D to the G. So when we look in the spreading map of this kind of the mutation, so we can see here that also already analysis by Korber et al. Recently, just August 2020 in a Cell journal, and he also presented here that the SARS-CoV-2 virus is circulating the human population globally, that now is becoming increasing of this mutant for the G, replacing of the D, and becoming the blue color is more infectivity is increased and larger in the population in the people in the world. So you can see here that the mutation and then becoming increasing the infectivity. But the pathogenesis and also the other effect of the disease is not reported yet. So we don't know the mechanism really happen in this kind of the mutation. But the increasing of this infection is becoming enlarging. So you can see here that the original of this kind of the point mutation is original come from the Europe. So why when we are designing the like phylogenetic tree, we always many kind of mutant or variant of the COVID-19 is come from the Europe. So when we have this kind of the mutation, so maybe the phylogenetic will be related to the strain of from Europe. So this is the original thing. When I'm enlarging this map, then we can found here. And Indonesia is the part of this area. So when you click the blue color, so you can check this is also the East Java now appear, and then the west of the Java. So we Indonesian data now is very, very limited that I mentioned before. Is only 21 virus already submitted in the GSET, and this kind of mutation is found around 8 data. 8 whole genome viruses from Indonesia, I think the west and the east of Java. And we are also found in Surabaya from infected patient, this kind of mutation. So now we have around 8 data of this kind of mutation in Indonesia. I think that this is so, it's really so very, very low information. Yes, this still limited information. So it's mean that Indonesia is need to analyze the whole genome sequence, and then we can submit it more data and also get more data about this mutation, because from other countries is already so many people infected with this kind of the mutant. So does a mutation matter and then also have the serious problem of this kind of the to the infected patient? So this is not really known about that yet. So the deeply research for this kind of the effect or impact of this mutation is still in process with many kind of researchers in the world. So mostly this kind of the mutation is doesn't matter, but however, in the case of the D mutate to the G, the glycine in the number of location is 614. So many publication journal in international journal is already explained that the D614G mutation in SARS-CoV-2 spike protein reduces the S1 shedding and increasing infectivity, and also transduction of multiple human cell type. And also this is the source, the journal that all of you can also download and read of this kind. And also the variant of this mutation in increasing effectivity and also retain sensitivity to antibodies. They target the receptor binding domain and also increasing the infectivity in H2 receptor dependent manner. So this is the publication regarding to this kind of mutation. So now becoming more attention, so many countries pay attention for this kind of mutation. So about in our university, what we already done for this analysis of this mutation, we already submitted 6. The color is red. So the 2 of them is very related to European virus strain, and we've found for the 2 this including the mutation of the D to the G. But the interesting one is the ID number with the end of is really interesting because it's not only have D640G, but also indicated as showing the other mutation is very interesting for us to analyze more detail. So when we look here, we see that the other mutations. So now we are in also writing a manuscript for this together with our team in Airlangga to explain is there any influences about this mutation and also this mutation together, separate and together. What happened if they are two or if they are only one by one? So this is we already now we also still in progress of this kind of the mutation. So for the second one that we interesting in doing the research in more detail about the other mutation is the Q to H, yeah, to histidine, and the location is 677 that already indicated in the GSET data. So you can see here why we are very interesting with this kind of double point mutation, because this is only the one in Indonesia that now this information is still only the one from Indonesia. This is from Surabaya, and this is also from our data analysis. So why we are very also interesting with this. But we don't know, maybe because our data is very limited. So maybe we can find this point of mutation in many kind of another city or another area in Indonesia. So why our research now is blocking the area of the mutation, so to make more faster information and then more easy to analyze the point of the 2 point of mutation in this information, so we can also give more information to from Indonesia to global data in GSET for this kind of only for this point mutation. So this is appear in March 3rd, 2020, and the last for before in the March 3rd we only have 6 countries. But now it's already become reached 99 infected patients. Even though the percentage is still very low, but this is interesting also to analyze in detail. So it's mean that the D to G is already started in this mutation happened in January, but this one is still in March. But now is 60,000, but this one is becoming 99. I think the both of them will be interesting to analyze. Why? Because we also be analyzed by in silico analysis with our Airlangga Universitas Airlangga team that we just mentioned to be published. We still now writing the manuscript because this mutation is very near with the profiling of the another protein, the furin cleavage site. This furin is another protease. But this is belong to the host, the human furin, not belong to the virus. So the furin, what is the relation between the furin and the virus? The relation is this kind of the furin will be cleave the S1 and S2 subunit of the spike, and then will be support or help the infection spike to bind the H2 receptor. So this is increasing the binding energy is higher than the wild type or without this kind of mutation. So we are only still doing analysis. Is this kind of the mutation will be give influence of the furin activity on S or not? We don't know yet until now. We still studying about this position of point mutation. So we don't know yet until now. So our team also already designing the modeling of the mutant. So we can see here, we have 2 mutant in one spike location. And this is in the loop area. This very near with the cleavage site of the furin, and furin will be help the spike to bind the H2 receptor, but we don't know yet the mechanism. So this is still our challenges to explore this mutation. So the other side is very important for us is how to develop the drug, new drug development for specifically for the this virus of COVID-19. So we know that the two protein in GZ will be the target of the drug. So we design this drug is a very new designing by synthesize the organic single compound to be active bind to the active site of the main protease. So there are two protein that's very interesting to be our target of the drug development is main protease and RNA polymerase. So this is the structure we already can get from the protein data bank, and we can see here this kind of the main protease is dimer. So when we open just one single site, so we can see here that this is the native ligand that binding in the active site of the main protease. So we then try to designing our synthetic compound with the single compound, and we can see we compare to with the drug antivirus is already in the market. So we can see here that the binding energy of our 5 compound is higher than the marketed drug is already sold as an antivirus, but not specifically for COVID. So this is our study also. We are also interested in this kind of information for the biomolecular of COVID-19. So this is the higher binding energy, and this is also done with our Universitas Airlangga researcher team, and we can see here that the active site of this enzyme in this site. So you can see here then how chloroquine bind with this kind of the active site of the main protease. And how about afigan? You know that this is I think recently also already mentioned by several information in the social media, and this is asperidine, and our compound, active compound, we call it Airlangga UNER1, UNER2, UNER3, UNER4, and UNER5. And now we started to focus in UNER3. Why? Because we already do the preclinical trial in vitro and also now still doing for in vivo analysis. So you can see here, this is the UNER1, and then the other one is UNER2. This is the third one is UNER3. This one is UNER3, so you can see the differences, and this the other is UNER4 and UNER5. So our research is still in continue. So then the other target is RNA polymerase. So you can see here the RNA polymerase is very important for the virus to amplify their genome, of course, but the main protease is very important to packaging the new protein like spike, envelope, and many things, the compartment of virus. So why drug targeted is this kind of two protein, but the vaccine is usually in the spike or any other target. So the RNA polymerase is very important, and you know that the remdesivir is already known. You already known all that this is now used in USA. So the remdesivir, if we look more detailed, we can check here. Is the remdesivir really a block or inhibit the RNA polymerase of the COVID-19? So we can see here in the Wanchow in the science paper, you can see here how this kind of the remdesivir block the RNA polymerase. You know the RNA polymerase you can see in the active compound, the meaning of this nucleotide ring. It's very similar with the RNA polymerase substrate. So we can then make the virus bind with this kind of the drug, so then they can stop. This is why we also designed the drug based on the binding energy with the two of protein. Thank you very much. I hope that the time is not more than 15 minutes. I'm sorry, Professor Tjandra. I hope that this is also very meaningful for us. Thank you very much. So the time I'll give to Professor Tjandra. Thank you.
Thank you so much, Prof. Nyoman. So, Prof. Nyoman already bring us to the various possible mutation, but not only mutation, but how it related to the treatment of COVID in the coming time. So, before proceed to the next speaker, I just would like again to remind the participant that you can raise your question, or you can write down your question in the Q&A button. If you click the Q&A button in the bottom of your monitor, there is a Q&A, and you can raise it there. So the presenter, could read that question and prepare for the answer. So now we are coming to the next speaker, or the second speaker. This is Ibu Vivi, of course. All of us already know, and she already even opened this international webinar. So Ibu Vivi now is the director of Center for Research and Development of Biomedical and Basic Health Technology. So it's difficult for me to read it in English because I know her designation in Bahasa. But anyway, so this is the English formal terminology. Director of Center of Research and Development of Biomedical and Basic Health Technology from NIHRD from 2019 until now. So Ibu Vivi has a lot of experience. I know her from the many years ago. She taking care of various quote-unquote pandemic or outbreak or epidemic happened so far in Indonesia, and she is the one behind the lab of NIHRD. So Ibu Vivi will deliver a presentation entitled, where's the title? Title is somewhere here. Let me find it. Indonesian challenges in the development laboratory network for COVID-19. Ibu Vivi, over to you.
Thank you, Professor Tjandra. Greetings to all participants. I would like to share some experience and best practice from Indonesia about the development of laboratory network for COVID-19. Yes. First of all, I would like to share the situation of the COVID-19 in Indonesia. Next, yes. Milestone of the laboratory network, challenges and best practices of establishing the laboratory network. Next. Indonesia is an archipelago country with 17,000 islands and around 270 million population, and large and diverse population with uneven distribution. We know all of the Indonesia have 34 provinces with population density around 115% per kilometer square. And also we have the high migration between island and cities, even between countries for tourism and also health pilgrims. Yes, next. Yes. From this slide, I would like to give some insight of the COVID situation in Indonesia, how the government of Indonesia respond to the COVID-19 threat, and can be seen from this timeline. Starting when the cluster pneumonia reported in Wuhan last year, Indonesia responds to establish the guideline for prevention and control. After the WHO announced as public health of emergency of international concern, we follow with the Ministry of Health decree for anticipation and control of outbreak. As we know, in March, Indonesia reported the first case in COVID-19 case, and for the anticipation of the increasing number of the cases, Ministry of Health of Indonesia announced the decree for the establishment of referral hospitals. And finally, on March 11, WHO announced the COVID-19 as pandemic, and at the same time, the president announced the presidential decree for task force to accelerate control of COVID-19. Yes. This slide also explains the progress of establishment of COVID-19 laboratory network, starting with the 12 laboratories in March 2020 and increasing to 45 and 49 before the end of March, followed by establishment guideline for laboratory PCR testing of COVID-19. And until recently, there are 295 molecular laboratories made for COVID-19 testing. And next. The composition of the 322 laboratories network under the Ministry of under some ministries like government laboratories, Ministry of Health, provincial and also hospital, laboratory under Ministry of Education, Ministry of Research and Technology, FDA Indonesia, and under Ministry of Agriculture, and laboratory of Indonesia National Armed Force or Police, and also private laboratories. And the Center for Research and Development of Biomedical and Basic Health Technology as National Reference Laboratory for COVID-19. And this is also strengthening previous decree. And we can see in this slide, we already have 322 laboratory testing for COVID-19 from the in all provinces in Indonesia, in 34 provinces in Indonesia. Next. And based on our experience until now, we face challenges on many aspects, like coordination. The laboratory under the lab network belongs to many different authorities. So this is also some challenge for the coordination. And also we understand the geography of Indonesia, that archipelago country, and need additional effort for the shipment, can have laboratory examination delay, and also lack of uniform information system for the recording and reporting. This is also the challenge for laboratory network. And need special laboratory technician, and also special laboratory design and infrastructure such as BSL-2 facilities, and also real-time PCR machines. So some challenges for laboratory network development. Next. The establishment of laboratory network needs also evaluation and also monitoring. To maintain the quality of the COVID-19 testing, we develop some activities such as external quality assurance, also training for PCR, and also biosafety and biosecurity, reagent validation, and also laboratory information. We also involve in research and collaboration, such as for COVID-19 vaccine collaboration, SEARO epidemiological study, and plasma convalescent clinical trials. Some of the activities like external quality assurance program, until recently, we received specimen from 153 laboratories out of 322 laboratories. Results of the evaluation can be seen from this table. And the range of the correct result from the laboratories are 60 to 100% of positive specimens, and 70 to 100% for negative specimens. This is the external quality assurance from 153 laboratories, and we will still receive another specimens from the other laboratories. We also conducted several virtual training collaborations with CDC, APHL, and also WHO. The first training conducted on 28 April to May, which covered more than 1,000 participants from many institutions in all of provinces Indonesia. Lanjut. Yes. Following the previous training, we also conducting on the job training to a lot of laboratories and hospitals with national standardized training modules. And we also developed the standardized training modules. And this is some picture on the job trainings activities in several laboratories in Indonesia, and also included Sutomo laboratories and Wahidin Sudirohusodo laboratories laboratory in South Sulawesi, and also in Denpasar. This is some on the job training activities, and the trainers come from NIHRD. Next, yeah. For the information of the data, we will explain about the development of laboratory data information system. This reporting using all record TC19. TC19 is tracking COVID-19 application, is an application that has been refined based on input from task force team, hospital, local health office, and also laboratories. The process, the Puskesmas, we call it Puskesmas or health offices, send the data to the laboratory, and also the laboratory will perform the examination, and the result will enter to this application. And the authority can see the result from this application directly on time. And then lanjut. The health facilities for the specimens collection from hospital, health center, health offices perform data entry, the patient identity, and specimen collection. After entering all data on the application, the specimens contain the patient identity data and specimen history can be printed from the all record applications. When entering the result of the application, the laboratory only needs to search by entering the NIC, the number or the identification code for the patients, and we can see, the laboratory can see and can input the results. Now, 4,714 health facilities in 34 provinces has been recorded and have account as a health facility of specimen sender to this application. Ministry of Health also conducted reagent validation since there are many reagents available in the market recently. We conducted validation for viral transport medium. Next. Also, we also conducted reagent validation for RNA extraction kit. And the other is for validation for RT-PCR kits. Yes. And some collaboration for COVID-19 vaccine also conducted with Biopharma company for Sinovac and recombinant, and also vaccine for recombinant viral vector vaccine with Sutomo Hospital, Airlangga University, and also Manchester University. Next. There is also collaboration with WHO together with many countries for seroepidemiological study. The objectives are to measure the seroprevalence of antibodies to COVID-19 in the general population by sex and age group, to estimate fraction of asymptomatic, presymptomatic, or subclinical infectious in population and by sex and age group. And next, the location of the seroepidemiological study in 17 provinces and all this is cross-sectional investigation. We also involved in clinical trial for administration of convalescent plasma as additional therapy for COVID-19. The objective is to measure know the efficacy of convalescent plasma therapy for people with COVID-19 in reducing mortality rates for COVID-19 sufferers in hospital. And also this collaboration of this study between NIHRD, Ekman Institute for Molecular Biology, Indonesian Red Cross, and also hospitals. The period of this study from June until December 2020 involving 30 hospitals in Indonesia. From this presentation, the future directions, we will maintain the maintaining and improving laboratory qualities, including biosafety and biosecurity, strengthen networking within laboratories, improving national laboratory data management system, and also strengthen collaboration on research and development on COVID-19. Thank you for the presentation, and back to the moderator.
Thank you, Bu Vivi, for giving us the overview of the lab network and the lab activities of the whole country of Indonesia handling this COVID pandemic and its challenges. Now we are coming to the third speaker, and also thank you, Bu Vivi, for the time. It's less than 20 minutes, so we can have some more time for discussion. So I would like again to remind participants, please post your question. I saw there are already 2 questions has been posted, and the Q&A one even already answered by Prof. Nyoman, but the other one will be for Bu Vivi. Now we go to the third speaker. This is Dr. Bahl, Dr. Sunil Bahl. Now he is a team leader, a regional advisor for IBD, Immunization and Vaccine Development in the Southeast Asia Regional Office of WHO here in New Delhi, so in the same city with me now. And then Dr. Bahl will present. We follow the one on the flyer. The title will be, let me check it. Vaccine development, policy and allocation, procurement and delivery. This is what he is doing now for to ensure how the policy and allocation, procurement and delivery will be done properly for the COVID vaccine. So Dr. Bahl has a postgraduate degree of Doctor of Medicine in Community Health from the National Institute of Health and Family Welfare, New Delhi, India. Dr. Bahl, over to you, please.
Thank you, Professor Tjandra, and good afternoon to all the moderators, speakers, and participants to this webinar. As has been mentioned, I will be speaking briefly on the COVID-19 vaccine development, on the policy and allocation, and the procurement and delivery of this vaccine. Now, on the before I get into the vaccine itself, I just wanted to introduce to all the participants the concept of the ACT accelerator, as it is called, the Access to COVID-19 Tools accelerator, which was launched in April 2020 by the Director General of the World Health Organization, the President of France, the President of the European Commission, and the Bill and Melinda Gates Foundation. What this initiative does is to bring together a range of partners to promote and accelerate the development, production, and the equitable distribution of COVID-19 vaccines, diagnostics, and therapeutics. And there are a number of key partners that include WHO, the Bill and Melinda Gates Foundation, the Coalition for Epidemic Preparedness Innovation, or Sp.P as it is called, Gavi, UNICEF, and so on. So there are a number of partners, including the private sector partners and other stakeholders that form this ACT accelerator. Now, today we are going to be focusing not on diagnostics or therapeutics, but just on the COVID-19 vaccines, which is one of the pillars of this ACT accelerator. So if we look at the vaccine pillar of the ACT accelerator, this pillar has 3 work streams. The first work stream is around the development and manufacturing of the COVID-19 vaccine, and it supports the research and development, as well as the expansion of manufacturing of promising COVID-19 candidate vaccines. And the lead agency for this particular work stream is the Coalition of Epidemic Preparedness Innovations, or Sp.P. The second work stream is around the policy and the allocation of the COVID-19 vaccine. And this work stream is led by WHO, and it primarily works to support the development of a global policy and an allocation framework on the use of potential COVID-19 vaccines. The third work stream is around the vaccine procurement and delivery, led by Gavi, and it is to support the availability of and the access to the safe and efficacious COVID-19 vaccine. So I'll be spending a few minutes on each of these 3, and then I will stop. So first, on the COVID-19 vaccine development and manufacturing. So there are, as of day before yesterday, that is 25th of August, there are 31 candidate vaccines that have entered the clinical trial phase. 6 of these are in the phase 3, and they are listed here. They include the vaccine being developed by the University of Oxford with AstraZeneca, the Sinovac vaccine, 2 vaccines from China, the Wuhan with Wuhan Institute and the Beijing Institute of Biological Products, the Moderna vaccine, and the BioNTech Pfizer vaccine. So these are the 6 out of the 31 that have reached phase 3. The rest are either in phase 1 or phase 2 of human clinical trials. In addition to these 31 candidate vaccines, there are 142 candidates that are in the preclinical phase, which includes the animal model studies that are being done on these vaccines. Now, these clinical trials that are being conducted on the 31 candidates are for not only the efficacy of these vaccines, but also on the safety of these vaccines. So both efficacy and safety of the vaccines is being tested as a part of the clinical trials that are being conducted. Now, in terms of the kind of vaccines or the types of vaccines that are being tested and for use, potential use, there are different platforms that are being used, and therefore there are different types of vaccines that are being developed. And these can be categorized primarily into 4 groups. You know, those that are using the virus vaccines. Now, the virus could be a weakened virus or an attenuated virus, as we call, or an inactivated or a killed virus. So there are weakened virus vaccines, or there are inactivated virus vaccines. And the examples of these are the Sinovac and the Sinopharm vaccines. The second group of vaccines is the viral vector vaccines. And the example of this is the Oxford AstraZeneca vaccine that is being developed. There are also nucleic acid vaccines. You know, it could be the RNA vaccines or the DNA vaccines. And the examples of these are the Moderna vaccine or the Pfizer vaccine that is being developed. And then the fourth group is the protein-based vaccines, and the example of this is the Allocation vaccine. Now, as you would imagine, when there are different platforms and different types of vaccines, these vaccines have different characteristics as well, and they differ in the number of doses that will be required, in the timing of the administration of these vaccines, when more than one doses have to be administered, also in the storage conditions of these vaccines. There are other differences as well in terms of the scalability, how fast we can increase the production of specific vaccines. So depending on the type of platform that is being used, these vaccines differ in that aspect as well. Now, these are just some examples to show you how they differ. I've listed a few of the potential COVID-19 vaccines that are being developed, and you can see different platforms that they're using. And then you see the number of doses, so there is a vaccine that will require just a single dose, but many others will require 2 doses and some even 3 doses of the vaccine. And when they require more than 1 dose, the timing of doses, the interval between the 2 doses could be 14 days or 21 days or 28 days, depending on which platform and which vaccine are we talking about. There are also, most of these vaccines are proposed to be developed to be given intramuscularly. However, there is one vaccine, at least the Cadila Healthcare vaccine, that is expected to be given intradermally. So this is just to show you how these vaccines are different from each other because of the differences in their platforms and the methodologies being used to develop these vaccines. So that's on the research aspect of the vaccine. Now, getting on to the second aspect, which is around the policy and the allocation of the COVID-19 vaccine. So once these vaccines are developed, how are we going to decide, who gets the vaccine and when? Because there isn't going to be enough vaccine available in the beginning, for sure. While there are many of these vaccines that are in the phase 3 clinical trials, there may be some failures as well of these vaccines. And even if some of them get through, it will take time to manufacture sufficient quantities of vaccines to be provided to the entire population. And therefore, a policy will have to be developed, and how these vaccines are going to be allocated is going to be an extremely important aspect that will have to be factored in. And as I said, WHO is leading the work on this. There are 3 components that are being worked on. The first component of the formulation of the vaccination strategies is the allocation framework. So this framework is expected to set the frame for the overarching goals and priorities for COVID-19 vaccine. So what are our goals and who are our priority populations for COVID-19 vaccine, independent of the candidate vaccines? Irrespective of which candidate vaccine comes, how do we decide what our goal is and which are our priority populations? That's what the allocation framework is expected to do. The second component is the strategic advisory group of experts that is helping to, again, decide on the policy and the allocation of vaccine, because this is going to provide guidance and policy advice in the context of specific candidate vaccines. So the first one was independent of the candidate vaccine. The strategic advisory group of experts is going to look at specific vaccines and then provide guidance on how the vaccine can be used and what kind of vaccination strategies will be required. Examples of this are, for example, there may be a vaccine that may be more effective in the elderly population or less effective in the elderly population, or not advised for pregnant women or not advised for immunocompromised people. So those kinds of specificities around the individual candidate vaccines will what the strategic advisory group of experts is going to be looking at, and then providing advice on how they be allocated for use when they become available. And the third aspect around the policy and allocation is around the regulatory, the safety, and the monitoring of these vaccines, to provide guidance on the regulatory issues, the safety and monitoring, both for the candidate specific and the system specific approaches. So those are the three components that are important from a policy and allocation of vaccine perspective that WHO is going to be providing guidance on. However, the final decision on the policy allocation and the vaccination strategy will be made by the countries based on the overall guidance that is provided. Now, just getting a little bit onto the details of the prioritization process, three priority populations and multiple potential target groups are being identified. This is still work in progress. However, it is clear that the populations that are at a priority must get a vaccine are those with higher mortality, serious morbidity rates than the general population, and where the risk-benefit analysis warrants a vaccination. So that's kind of one group. The other priority population is individuals who provide critical societal goods and services. So there could be people who are working in the health sector or are in the front line of other sectors, social sectors, and therefore will require a vaccine on a priority. And then the third group is those individuals who are more likely to be exposed and spread the virus. For example, those that are exposed to a super spreader event, if there is one in the country. There are examples of the target groups. The target groups could be the elderly, which is more than 65 years, workers in the health and social care settings, those less than 65 but having comorbidities, other essential workers, adults in densely populated areas, and then the rest of the adult population. So the prioritization of the target groups who we would want to vaccinate are defined by common characteristics such as age, health status, occupation, to help to identify them. Now, the allocation framework that is being developed has a goal, and the goal is to protect public health and minimize societal and economic impact by reducing COVID-19 mortality. So that's the goal which with the allocation framework is working. So the real goal is to minimize the societal and economic impact by reducing the mortality due to COVID-19. And with that goal, then the prioritization gets a little easier, because if that's the goal, then who gets the vaccine first is the health and social care workers. And that's what is being proposed as a part of the allocation framework, that once the vaccine becomes available, countries start receiving the doses proportional to their total population to cover 3% of their population initially, which is roughly the number of health and social care workers in a country. And then when further tranches are made available, the next group of risk category, which is the high risk adults, start getting, and this could include the elderly population, the adults with comorbidities, or others, depending on the locally relevant risk factors. And this will take the whole population, requirement of doses to 20% of the total population. So it is proposed that once the vaccine becomes available, it is distributed in proportion to the population of the countries, first the 3% of the population, and then subsequently going up to 20% of the population. Of course, when more vaccine becomes available, then it goes to the others beyond the initial 20%. It is also proposed to have a buffer to be set aside for emergency deployment if there are any immediate needs for the COVID-19 vaccine. So the key features of the allocation framework are that it is a proportional distribution. So it is proposed to develop 2 billion doses, which are allocated proportionally to population, to both the countries that are going to be self-financing and those that are going to be funded, keeping a buffer of 5% for emergencies and acute outbreaks. The vaccines are expected to be rolled out as they are produced until the countries reach their indicative target amounts. And then the final key feature is that the country policies will guide the national priorities for vaccine use. WHO, of course, will provide recommendations based on the SAGE advice, which will support the country deliberations and decision-making process. So that's on the allocation and the policy issue. Now, the last is around the COVID-19 vaccines procurement and delivery. So how is the procurement going to happen and how is the vaccine going to be delivered? And for this, at the global level, there is a COVAX facility or a COVID vaccine access facility, which is a global mechanism to pool the resources and the demand for COVID-19 vaccines with the objective of accelerating the availability of and the access to safe and efficacious vaccines. So the facility, the COVAX facility, it kind of pools the demand on one side, so it consolidates the country's buying power and provides the countries access to a broad and actively managed portfolio of COVID-19 vaccines. On the other side, it has the manufacturers which are going to be supplying the vaccine. So it pools the supply on the other side. It provides the manufacturers access to a massive demand assured market. So it's kind of pooling the demand to the supply of vaccines. And in order to ensure that this facility is operationalized well, countries have been categorized into two categories. They are self-financing countries, which is mostly the high income countries and the upper middle income countries. And the second group is the countries that are what is being called to be covered under the COVAX Advanced Market Commitment, which is a financing mechanism to ensure that the low income and the lower middle income economies, as well as the other IDA eligible economies, have access to COVID-19 vaccines. So the facility aims to make available up to 2 billion doses of COVID-19 vaccines by the end of 2021. Of these, nearly half to be financed by the advanced market commitment for the low income and lower middle income economies, plus the other IDA eligible economies, and nearly the remaining for the self-financing countries, which includes the upper middle income countries and the high income countries. The COVAX facility allows the countries to secure access to vaccines. As I've said earlier, the portfolio of vaccines is very large. We don't know at this point which vaccine will go through and will succeed and will prove it is efficacious and safe. So it is basically trying to ensure that there is a risk sharing mechanism. So it reduces the risk for the countries that are concerned about failing to secure access to a viable vaccine. And it is also reducing the risk for the manufacturers about investing without an assured demand. So it's kind of a risk-sharing mechanism. All these self-financing countries are being invited to be a part of this facility by making a commitment to purchase the doses and then also contribute funding for the procurement. The participating self-financing countries will receive access to vaccines produced by the facility at a negotiated price. On the other side, there are these COVAX advanced market countries which will get manufacturer-specific contingent volume guarantees and a market-wide demand guarantee for the manufacturers. So the lower income countries and the lower middle income countries participation will be supported by the ODA funding. As of 24th of August, there are 172 countries and multiple candidate vaccines that are engaged in the COVID-19 vaccine global access facility. There are 9 Sp.P supported candidate vaccines that are a part of the COVAX initiative. And there are another 9 that are under evaluation, and procurement conversations are ongoing with additional producers that are not receiving R&D funding through the COVAX. There are 80 potentially self-financing countries that have submitted non-binding expressions of interest to the COVAX facility. And there are 92 countries beyond these 80 that fall in the COVAX advanced market commitment that are eligible to be supported through the COVAX facility. Now, so that's on the procurement and the supply. Now, in terms of the last piece, which is the readiness of the countries and the delivery of the vaccine within the countries, there is work going on there as well. There is a global coordination mechanism that has been put in place, which you see right in the center of the circle that I've shown there, that is coordinating the whole process of country readiness and the delivery of the vaccine. There are pieces that are around this coordination mechanism, and that's what the project management of this country readiness delivery workstream is coordinating. These include the communication, the advocacy, and the training on the data and monitoring, as well as on the implementation and guidance. So there are technical areas, around the vaccine introduction, which includes the readiness assessment, the deployment, and the vaccine use. On the demand, which includes the acceptance and the community engagement and such like things to ensure that there is public confidence in the vaccine. And then there is the supply and logistics, which looks at the supply distribution and the logistic strategies and the cold chain that will be required. So this is a comprehensive kind of a work that is happening at the global level. This is also being translated at the regional level and at the country level. And what is expected at the country level for the deployment of the COVID-19 vaccines is the initiation of the development of preparedness plans for the deployment of a future COVID-19 vaccines. And there are a number of components, the prioritization of the population groups in the country, as I just described before, on the mechanisms and the platforms for the delivery of vaccine, because if the groups are adults or health workers, then the EPI platform for delivery of vaccine is not going to be enough. So there will have to be consideration given to additional mechanisms and platforms, the health system requirements in terms of, the human resources, the cold chain requirements, the capacity of the human resources, etc., will have to be factored and looked into. The regulatory pathway requirements, because if the vaccine becomes available, there will be a licensure of the vaccine required, and that will have to be also considered and decided now. The vaccine safety monitoring mechanisms, because this is a new vaccine, we will have to be ensuring that there are mechanisms to assess the vaccine safety post-marketing as well, in addition to the safety testing that is happening as a part of the clinical trials, the communication strategies to ensure that the vaccine is accepted and there is demand and confidence of the public in the vaccine, and finally the monitoring and the evaluation of the use of the vaccine. So these are some of the key aspects of the vaccine preparedness and the readiness that countries are now beginning to do and will take shape as we go forward. So that's pretty much on the three components that I was asked to speak on. Over to you, Professor Tjandra. Thank you very much.
Thank you, because I'm also, again, mute by the committee, but now it's unmute. So let's go directly to the question and answer. So Dr. Bahl, you may read the question and answer. There are several question and answer question to you, even though some are in Bahasa, but I can translate later on to you. Let's begin with Prof. Nyoman. You have 3 or 4 questions. I know you already answered the first one regarding the impact or the relation between mutation and vaccine. So Prof. Nyoman, over to you. Prof. Nyoman, are you with us?
I'm sorry, Prof. Tjandra, because the organizing committee just opened the video and also unmuting for the speech. Yeah, so sorry. Yeah, I have. I think I've already read one regarding to the mutation and related to the vaccine. So if we look for the 2-point mutation of the D to G 614 and also Q to H for 677, until now we don't know yet the pathogenesis and related of this kind of the point mutation. But the location of the mutation is in spike. We are already analysis, still only in silico analysis, docking and also modeling. So we also already designed this kind of the point mutation as our protein recombinant to compare with the wild type from the Wuhan. So it's mean that this research is still in progress, but we designing the variant of the D, G, and Q and H to be our other protein recombinant for vaccine candidate. But this variant is only used for comparable study with the wild type, spike, and also wild type of the RBD, the receptor binding domain. That is the answer, Professor Tjandra.
Actually, you still have several questions. If you go into Q&A,
Yeah.
so let me read it for you. The question is regarding the last question just now. Why we have mutation? Is it because we do not control it quite well? But I think you can mention that mutation is happening everywhere.
Yeah.
The other question is regarding the, is there any relation between severity of the patient with the mutation? Is it mutation then related with hypoxia, for instance? And
Yeah,
there
I
is
think
also
mutation,
question
yeah.
of what is the treatment for severe COVID? Those are the
Yeah.
4 questions.
I think virus is the virus is very the one of the smart organism that we know that because the virus it's doesn't have any fidelity or proofreading in their genome, so they will have their amplified their genome in the host. Of course, the host will be also different if we know that this is like a zoonosis from the animal to human. But this is the smart of the virus. So they can adapt it with the host. So why they make a mutation effect to have like more infectivity to the host. So to adapt with the kind of the host environment. So this is, I think, the virus itself is very, very smart. So we don't know when and then why the viruses make a mutation. But the important thing that the virus need hosts to amplify their genome and also to design and packaging their own protein, and also they package their body to infect another host. So why this is will be more very high spreading until now. So we difficult to control the mutant because this is very natural happen with the virus. So we hope that even though now we find so many mutant, but they still doing just only for the infectivity, increasing the infectivity. We hope that it's not related to the pathogenesis of this virus to the host. We hope that. But if the amount of the virus is very, very high, so I think this is also will be another problem with us. I think that's Professor Tjandra.
Yes. Okay. So there are still 2 or 3 questions, but let me just compile it.
I think for the drug combination, if I'm not mistaken, I show.
Yeah. If I can read it for you.
Yeah.
So the question is about what is the drug combination for COVID-19 with comorbid?
Yeah, because we didn't do for the combination of the drug. So what we develop is really a new designing as anti-main protease that we are designing by synthesize a new single compound as a drug candidate. So why we call it drug development in our university now? We still doing in that. So for we have already designed 120 single compound, and then we check by docking to the main protease that we already that I already mentioned in the video, the short video, how the single compound, we call it inhibitor, bind with the main protease to block this kind of the main protease to do its job to the viruses, to help the viruses. So we block that kind of the active site. So it means this is single compound. Now we already done for the in vitro analysis, and now we started entering in vivo analysis. So it's the drug discovery or development is still need time, maybe until 1.5 years next, because it's not, because this is a really new synthetic of organic active compound. This is we call it in bahasa Indonesia, Prof Tjandra, is OPB ya, obat pengembangan baru. Mungkin Prof Tjandra, I think Prof Tjandra know in Indonesia we know that the OPB is a new drug development, so it's need preclinical trial and also clinical trial. So it's mean we still need more time. So I think not this year, maybe next year. So because we still do, we just finished for in vitro analysis. Now we do for the in vivo analysis. So that is not combination. This is single compound. I think this is the answer for Tjandra.
Thank you, Prof. Nyoman. Actually,
Yes.
there is a question, but we only have 20 minutes, so we already passed the 7 minute or 8 minutes. Now we go to the next 7 minutes for Bu Vivi. You have 3 questions. I think you have read it, but anyway, Bu Vivi, over to you. The question is about what effort should be made to restore public health condition amid COVID-19 pandemic. I think the committee should unmute her.
Yes. Thank you, Pak Tjandra. Thank you, Prof. Tjandra. For the public health concern, we have several response for public health concerns. The first is for the diagnosis, testing of the specimens appropriately, and all of the suspected cases have to be tested by PCR. And then we can, the next step is to educate the people or community how to use the mask and for the cuci tangan. Hand wash and also physical distancing. That's for the education for the community for the prevention or for the COVID-19 transmission.
Yes. And the second one is about the VTM and PCR. What will be the advantage and disadvantage of each of them? Positive as well.
For the testing of validity of the reagents, the questions?
The VTM and the PCR.
Yes. The validation result of VTM PCR reagents. Yes. For the VTM, we have several kinds of VTM. One is VTM is only saline, and the other is VTM with lysis buffer. The VTM with lysis buffer, some of the VTM makes results of invalid results. So we have several results for the VTM validation, and we think that it's better without the lysis buffer for the VTM, since the shipment of the specimens needs some time from the remote area or from the district to the lab, or from the hospital to the lab. So it will be the RNA is not good if we use the VTM with lysis buffer. And for the PCR reagent, we have several kind of PCR reagents in Indonesia now, but we are testing that. And we have the sensitivity and specificity of PCR reagent is around 80% until 100%. And this is very broad result of the PCR reagent. So better we use for PCR reagent with 100% of sensitivity and specificity. Yes, thank you, Pak
Okay,
Prof. Tjandra.
if you have two more questions, and the last one just came from colleague from BPK, basically. So the two questions for you. The first one is to see the number of cases are increasing in the country. Do we need to do the PSBB again? I know it's not lab, but since you represent Ministry of Health, you may like to reply. Do we need the PSBB again because number of cases are increasing?
Yes, Prof. Tjandra, but the question is from Padang, I think.
Yes,
Stickers,
exactly.
yes. The PSBB is limitation of the limitation from, maybe it's local authority local authority policy. So now is we are not using the PSBB anymore, but we have to several kind of activity or to prevent, so like using the mask and also hand washing and also the physical distancing.
Okay, so
Back
you
to
still
you,
have,
Prof.
you still have several questions, but if you could kindly just reply to this question from a colleague from Badan Pemeriksa Keuangan. So how many laboratories should be ready to support the government effort to end this pandemic? And the government already set a target. When will the pandemic should be end? But yeah, at least target of lab maybe you can share before we move to the next speaker.
Yes, the pandemic, how to, when the pandemic will be end? Maybe Prof. Tjandra, you can also help.
No. I mean the first part of question: how many laboratories should be ready for supporting?
Okay. Now is we have already 322 laboratories in Indonesia, all over Indonesia, 34 provinces. So we have to increase the, not increase, but strengthen the laboratory capacity from the testing and also from the infrastructure and the establishment or distribution of the reagents on time. So we have to also to maintain the quality of the laboratories. This is very important to maintain or to strengthen the laboratory in Indonesia to test the specimens in time, so not have a delay, so we can have the results good and deliver the result to the patients, to the cases, no delay. That's all, Prof. Tjandra.
Thank you, Bu Vivi. I know there are still several questions and also 6 or 7 people raised their hand, but let's go to Dr. Bahl. So at least one is in English, you can read. So the question is regarding protection.
So I have already looked at 2 questions in English. First one is on the subpopulations that should be included for vaccination. And so I think, as I mentioned, the decision on the subpopulations that should be included will have to be based on the data in the country. So the epidemiology in the country, the transmission dynamics, and the risk of the mortality will have to be assessed, and based on that, there will have to be a decision taken in the country on which subpopulations are at a higher risk. Of course, SAGE is also looking at this issue and will provide some overarching guidance. There is modeling work that is going on as well that will be presented to the SAGE in early October, based on which SAGE will be providing its guidance. However, the final decision, as I said, will have to be taken by the National Immunization Technical Advisory Group based on the information within the country. So that's on the subpopulations to be included. The second question is interesting. It says, is there a guarantee that once somebody takes COVID-19 vaccines, there will be no COVID-19 vaccine? No, there can never be a guarantee. There is no vaccine ever produced for anything in the world that is 100% effective. Every vaccine has an efficacy of 80%, 60%, 70%, and so on. So there is never a guarantee that 100% of the people who receive a vaccine will be protected. So depending on what efficacy is of the vaccine that is used in a particular area or in a country, that percentage will probably be protected. The rest will not be protected with the vaccine. The second aspect here that I would like to also highlight is that we still don't know what will be the duration of the protection because of the vaccine. So since this is a new vaccine, this is just coming in, will take some time to understand how long the vaccine provides the protection, and therefore it doesn't mean that if once a person has taken the vaccine, there is a guarantee that the person can never get COVID-19 vaccine. So we'll have to be taking that into account as well. The rest of the questions, Professor Tjandra, I can't understand, so you might have to.
So there are still some, but let me just take one. The question, one of the question mentioned that, so there are several candidates, as you mentioned, Sinovac, Moderna, etc. So how do we know which candidate will be, matched with COVID situation in Indonesia?
Well, we don't know. We don't know that yet. And that is the reason why the COVAX facility becomes important, because we don't know which of these vaccines will be effective and safe. Normally, whenever a new vaccine is produced and when the vaccines reach the phase of clinical trials, a large proportion of these vaccines fail. So anything between 80 to 90% of the vaccines fail. I'm not saying that that will happen with the COVID vaccine as well. There may be more success with these vaccines. We don't know yet, but we don't know which vaccine will be effective and safe. And therefore, I cannot give an answer with guarantee today that Moderna will be effective or Oxford will be effective or Sinopharm will be effective. And that's why, there is a risk. So if a country goes with a particular type of vaccine and, kind of assures that that vaccine will be, is what they would want. And if that vaccine fails, the country will be, in a difficult situation, because if they are banking only on one vaccine and that vaccine fails, they will have no vaccine. Whereas if there is this COVAX facility, as I said, is a risk-sharing mechanism. So if one vaccine fails and another vaccine succeeds, then the country will have access, all countries will have access to that particular vaccine that will succeed. So that's kind of the way and the risk mitigation that the COVAX facility is doing.
Thank you, Dr. Sunil. So colleagues, participants, so I think we have come to the end, and even more of the time allocated for this session. So thank you very much for the three speakers, three presenters, Prof. Nyoman, Bu Vivi, and Dr. Bahl, for their extensive, for their very impressive presentation and discussion as well. I know there are still so many questions, and even somebody raised their hand. So maybe the committee could compile all of the questions later on, and then perhaps find a way to reply that one. Let me just read three conclusions from each of the speakers. So from the first speaker, I think the main important issue is mutation has already happened, but it happened not only in COVID, but as well as other viruses and even bacteria, so like many other. So we have to we have to watch vigorously, intensively of this mutation. And as of now, we do not have clear information regarding the impact of mutation into the vaccine or into the medicine for treating COVID. And from the Vivi is kind of assurance that Indonesia is continuously expanded their high-quality laboratory capacity throughout the country, despite the, Indonesia has a wide coverage area, difficult geography, etc. But Ibu Vivi share with us the Indonesian capability to diagnostic with a high quality level. And the one that I prepared for Dr. Bahl is even after vaccine is available, then we have to find an extensive way, monitoring closely how to deploy it, how to making an allocation framework, how to delivery, etc. But the last issue raised by Dr. Bahl is we still have to wait for the result of the clinical trial so far that happened in several countries. So thank you very much again, all of the speaker and all of the participants for the first session of this international webinar. Thank you, and back to you.
Thank you, Prof. Tjandra, for nicely moderated these first sessions and all the three speakers, Prof. Nyoman, Ibu Vivi, and Pak Sunil Bahl. Now we would like to jump into the second sessions. And for these sessions will be moderated by Dr. Siswanto, Master of Health Policy. He is a senior health policy analyst and former DG of NIHRD. So he is also the member of the National Research Council and a member from the Committee of Komisi Keamanan Hayati and Produk Rekayasa Genetik National Committee in Indonesia. Pak Sis, if you already can listen to my voice. Yeah, Pak Siswanto.
Yes.
Okay, yeah.
Yeah. Thank you, Bu Uli.
Yeah.
So, good afternoon for everybody, especially for the speaker, also for the attendee of this webinar. So I would like to continue the task of Prof. Tjandra as a moderator, because Prof. Tjandra seem to have another video conference, something like this. So after we have a deliberate discussion regarding the global genomic sequencing of SARS-CoV-2 or COVID-19 virus, and then also the experience of Indonesia in improving the laboratory network for COVID-19 testing. And the last is how the progress of the COVID-19 vaccine in the world, and how we have the fairly access for each country to that kind of vaccine. Now we move to the second session of the webinar. First, we have Dr. Novilia Sjafri Bachtiar from Farma. So we can see in the screen the short CV of the Dr. Novilia Sjafri Bachtiar. So basically, she from Pajajaran University and now working in the Biopharma. Also lecturer in the master program of tropical medicine, and now also the WHO temporary advisor for MISA laboratory, because we have MISA laboratory, and one of the reference for MISA laboratory is also in Biopharma, apart from the NIHRD, and also as an authorship, a number of publications, also some books about vaccine development. So Dr. Novilia Sjafri Bachtiar will talk about challenges and progress in clinical trial of COVID-19 vaccine in Indonesia. As far as I know, that kind of vaccine basically is collaboration between the government of Indonesia and the Sinovac from the China. So please, Dr. Novilia Sjafri Bachtiar, so time is yours. So again, for the attendees, if you have question, just write your question in question and answer facility, not in chat facility. So again, in question and answer facility. Please, Dr. Novilia Sjafri Bachtiar.
Okay. Thank you so much, Pak Siswanto, for the introduction. First, I would like to thank NIHRD Balitbangkes for the invitation for Bio Farma in this opportunity. Greetings to the head of Litbangkes, Ibu Vivi, moderators, and other speakers. According to the organizing committee, I have to deliver about the challenges and progress in clinical trial. So this is really a very, very good title because actually we have conducted more than 30 trials, but this time during this pandemic is really a challenging one. So this is the outline of the presentation. The first one is about a biopharma approach for the COVID-19 vaccine. And then clinical trial in general, challenges in pandemic situation, and update on the clinical trial. In this slide, I would like to show you about our long-term approach for the vaccine development, not only for the vaccine development, but also for the capacity building. There is the National Consortium with the collaborators from the Ekman Institute, Bio Farma, and also NIHRD Balitbangkes. And hopefully, the first clinical trial will be conducted next year in Q3. And our short and mid approach for the vaccine development is a collaboration for formulation and filling of the COVID-19 vaccine with Sp.P, the Coalition for Epidemic Preparedness Innovation, but it's still in progress. And also with Sinovac, that maybe you all already heard from the media that we already start the clinical trial in collaboration with Sinovac, where Sinovac developed the inactivated vaccine and already finished with the phase 1 and phase 2 trial. So this is the general trial, the clinical trial in general. We can see here that we should start with the vaccine research. Usually in the common situation, vaccine research will take time very long, between 5 to 10 years. But right now during the pandemic, only 1 to 2 months, many research institutes already find the seed and go for the preclinical trial. And if the preclinical trial research is good, we can move to the clinical trial, start from the phase 1, phase 2, phase 3, and go for the registration to Badan POM, and later conduct the phase 4 clinical trial or post-marketing surveillance. Of course, all these phases should be conducted with good clinical practice and good laboratory practice. Talking about the challenges in the clinical trial, as we can see here, there is a potential accelerated pathway for COVID-19 vaccines. So we should conduct all the phase 1, phase 2, and phase 3 clinical trial, but some of these phases can be conducted in parallel. So after conduct the phase 1 for a several time, and then later we conduct the phase 2, and after the phase 1 finish, we conduct the phase 3 in parallel with the phase 1. This is just an example for for the potential accelerated pathway in a clinical trial. We know that Badan POM also facilitate the accelerated pathway by using the emergency use authorization and WHO by emergency use listing. So hopefully the vaccines and medicines will go for registration as soon as possible. Challenges in choosing a clinical trial center, especially in this pandemic situation, we have to implement the prevention protocol, not only for the investigators, but also for the subjects. So we have to make the SOPs to conduct the trial in pandemic situation. For example, we cannot invite so many people in the same time. We should provide the hand sanitizer, provide the masker, and so on. The other challenges that actually this time we have several clinical trials that should be conducted in several cities for other vaccines. For example, for the typhoid conjugate vaccine and for rotavirus vaccine. But most of them suspended their clinical trial activities because of this pandemic. So this is not easy to choose the clinical trial center. Of course, we have to choose the clinical trial center with experience in vaccine clinical trials. Challenges in logistics and transportation. During the pandemic, we know that very limited public transportation, especially between city to city. Many flight stop, no schedule for a regular flight. So it's difficult for us to send any logistic or even vaccine to other city. And there is a travel requirements to use the public transportation. So it's not easy also for us to do monitoring or inspection to a clinical trial in other cities. And also for the transportation, especially for vaccine will need the cold chain. So if we use the regular transportation during the pandemic, there is a risk to be canceled or delayed. And our vaccine also have a risk to have a higher temperature. We know that this vaccine is very limited, so we have to be very careful in during the transportation. So this is the challenges in communication and media. We have to be very clear with the media, and also we have to be very careful because sometimes we don't understand, and their statement in the media make other institution or other people in a difficult situation. And also so many interviewing agenda for this clinical trial. So we are so busy with the interview, but we also have to be focused on our trial and our scientific part. So and then we delegate our interview with spokesperson. Of course, we have to train them before they speak to the media. And the most important thing also, we should conduct the media briefing where we can train the media about the clinical trial, about vaccine, what is the regulation, what is the GCP. So they understand how to write about clinical trial. Other challenges that also often come to us is about the politics, about so many institutions also wants to be part of the trial. Many people, maybe a public figure, wants to offer the contributions for the clinical trial, but we have a protocol that we should follow, and then sometimes find lots of comments from other people, from public figure that not really understand about what is the clinical trial, what is the vaccine. So this is challenges and sometimes made our position more difficult. So the next presentation is about the updates on the clinical trial. So this is the collaborators between Pajajaran University and then NIHRD. Thank you for Ibu Vivi. So we have collaboration to do the neutralization assay for the serology in this trial. The vaccine is from Sinovac, inactivated vaccine, and Bio Farma as the sponsor, and also will do the ELISA SI. This slide just want to show you that we prepare this trial in a very, very short time. We start with May 6th, the meeting organized by National Institute of Health Research and Development, Balitbangkes, and then several meetings follow the first meeting. And in June, also several meetings with other institution. And as we can see here that the vaccine was arrived in July 19, and we got the ethical clearance in July 27. And in this pandemic situation, Badan POM allow us to do the submission for the clinical trial parallel with the committee ethics. So that's why we can get the Badan POM approval in the same day after the ethical clearance was issued. If you see the landscape of the COVID-19 candidate vaccine from WHO, we can see at the second row, Sinovac and the phase 3 study. If you click below link, so you can see this Indonesian registry for this clinical trial, collaboration between Bio Farma, Pajajaran University, Balitbangkes, and Sinovac Life Science. We also register our vaccine in our trial in clinicaltrial.gov. This is the link. And the update of the clinical trial, we start the trial for the first enrollment in August 11, 2020. Maybe some of you know that the president visit us in this first enrollment. Then follow for the next enrollment in August 14, and totally it's about 110 subjects already injected, and hopefully this week we will enroll another 150 subjects for the clinical trial. Thank you. That's all, Pak Siswanto.
Yes, thank you, Ibu Dr. Novilia from Bio Farma. So I think this is a good presentation in the sense that it is the real practice of vaccine clinical trial in the pandemic era. Even though it is in the pandemic era, but all requirement in the trial, like scientific perspective, also ethical perspective, also regulatory perspective, should be fully met. That's the important point. Now we move to the second speaker. The second speaker is Pak Ahmad Handoyo Utomo, Ph.D. He will just you can you can see the screen for the short CV. Yes, takes the Ph.D. in biomolecular medicine. So, and then now is also in a number of research involved, like stem cell and stem cell. I mean, and then also head of laboratory in Calbe Genomics. So basically very experienced in biomolecular, and he will deliver the topic, the overview of next generation vaccine platform for COVID-19. So I think it is a very important topic, especially for vaccine researcher, to find the another opportunity for establishing COVID-19 vaccine. So please, Pak Ahmad Utomo, now the room is yours. Thank you.
Jay, Dr. Siswanto for a kind introduction. So let me share my screen. Okay. Right. So thank you for the organizer to inviting me and share with you all regarding the next generation vaccine platforms to stop this pandemic. Yeah? Okay. So we know the COVID-19 has claimed hundreds of thousands of deaths, close to about 6 million to date. It's a huge number of cases, and it's in a way it is a bit ironic because we know a lot about the coronavirus, but apparently it's not good enough to stop the pandemic. The question would be why COVID-19 spread so fast and how different it is from SARS and MERS. We know both SARS-CoV-1, the causing agent of SARS, and SARS-CoV-2, causing agent of COVID-19, they both bind to ACE2 receptor. But yet, why it's so different? And that's because apparently the SARS-CoV-2, the spike domain of the SARS-CoV-2 actually has 10 times higher affinity, so it can bind more tighter, in spite of the 20% difference in genomic sequence. So this is very amazing. They're so different, but yet they can bind the same receptor. But also, it's also interesting why there's no vaccine against SARS and MERS, and that's because SARS and MERS are easy to be recognized. The people who've been infected with the virus, they have these symptoms, easy to detect. They're flu-like, they have a cough, fever, and the viral load actually it's within the symptoms window. So that's why it's very easy to isolate the infected person. But this is not the case with the COVID-19. What happened with COVID-19, this virus, though they bind ACE2 just like SARS, apparently they're actually more prevalent in the upper respiratory organs instead of in the lower respiratory organ. So when the virus actually replicate here, the patients or individual, they don't show the symptoms, but yet they can actually spread the virus around. And if you can see with SARS, actually it's the opposite. It's mostly actually replicate in the lower respiratory organs. So this problem actually becoming an issue. So how to stop the pandemic? So we have several options. One, how to flatten the curve, maybe through lockdown, just like the Beijing style, the Chinese style in China. Or we can do the massive PCR testing, the 3T, the test, trace, and treat, just like the Korean ways. Or possibly is the vaccination, or it could be a combination of 3 approaches. Now, right now, the world is not ready, and we do have some issues. We try to have several mobility restrictions, but without vaccination, it's going to take a while. It looks like it's never ending. But hopefully with vaccine, there's a possibility that most people will be vaccinated in a relatively short time and probably will stop the transmission. Now, but in order to address this issue, I have actually 2 outlines. One is how to make the vaccine fast warp speed, because everybody wants to go out, nobody wants to be stay at home anymore. And the second point is how to prove that it works. So let's see what kind of technology is available to us at the moment. So this is actually the classical method how to produce vaccine. You have to have a virus seed. So if you remember in the avian flu, when Indonesia was hard hit with the avian flu virus, Ibu Fadila Supari makes sure that this virus actually stay within Indonesia, not exported outside. Because the concern is that if the virus actually is exported outside without knowing, then the vaccine developer can make vaccine and sell it to us at a much higher price, although Indonesia actually is in dire need. So this is actually the classical way how to develop vaccine. You need the virus. You need the virus isolate. But now with this revolution in molecular biology, definitely you don't really necessarily need the virus. What you need actually is the genome. So with this genome, you can actually do this genetic engineering, and then you can design the vaccine what you want. And the step is actually much simpler. You don't need this very long and tedious work, but it requires two things. One, you need data transparency, and the Chinese has been quite kind to actually deposit the sequence right away within month of January, just a month after the pandemic broke out in Wuhan. And also you need global scientific cooperation. So these two ingredients are key if you want to get this vaccine to go up to speed. So I divide this cartoon into two. On the left side, you can see the classical platform. Actually, where the vaccine has been known to work. For instance, you can see the virus-like particle. Actually, it's been successful to develop a cervical cancer vaccine, the HPV vaccine. And also, if you notice, the Sinovac and Biopharma actually is doing the clinical trial using whole inactivated virus. But there's also several next generation platforms that is using the viral vector, using the adenovirus as a carrier. You have a DNA vaccine. I think Genexin from Korea is working with Kalbe, hopefully maybe at the end of this year. The RNA vaccine by Moderna and others. So if you look, there's actually some advantage and disadvantages. For the DNA vaccines, interestingly, since you're actually adding DNA directly, so to the human, what we work actually is the integration into human genome, which may cause cancer. So this is actually one of the formal worry that we have to think about. But also in Indonesia, the question will be whether it's halal. So any vaccine that requires cellular manufacture or cellular production, then there is actually some worry. Maybe there's some porcine component during the processes. So I think it's important to educate and actually to make sure that all the stakeholders understand what's going on in the vaccine production. Now, of course, the point of having vaccine is to develop immunity. The idea is to develop these antibodies and also to build the cytotoxic T cell so it can recognize the infected cells. Now, can this classical and next-gen vaccine induce immunity? And the answer is yes. All the platforms, whether they're virus with the virus carrier, the adenovirus carrier, whether it's a nucleic acid RNA or DNA vaccine, or the viral vector, they all can develop immunity. That's not a problem. But the question is, will it work? So right now we're in the middle of the critical phase 2 clinical trials. But there is a problem that we have to understand. The problem with the evidence-based medicine, we need to see data. I'm taking this an example using the plasma therapy, convalescent plasma therapy. The idea is quite simple. You take the plasma from a person that has recovered from the disease, from COVID-19, get the plasma out, take the antibodies, hopefully maybe some antibodies there, and inject it to a patient. Very straightforward. But the problem with the clinical trial, you need to get enough patients, enough groups in order to make a comparison. Now, unfortunately, in China, when it's actually done in China, the containment has been successful. So what happened is they cannot find patients. So when you don't find patients, how can you do clinical trial? So unfortunately, this is actually one of the papers saying that the convalescent plasma therapy is not really having conclusion whether it's actually effective or not. Now, back to vaccination. So when it comes to the vaccination, there are two aims. Can it actually prevent a severe disease post-infection? We know that many of the patients died of the pulmonary failures, they cannot breathe, very severe hypoxia, or whether they can actually prevent transmission and stop infection altogether. So in order to do this, since we are still waiting for the clinical trial, many vaccine developers used this monkey as a model, whether they can show some prevention of severe disease. So whether the monkey will get the disease after a vaccination or not. So they compare the vaccinated ones versus placebo. So let's look at the Sinovac, the inactivated virus, a classical platform. Yes, it can actually protect the monkey, the vaccinated monkey, from this severe pneumonia. So the vaccinated one, apparently they are protected quite good. And also in the RNA vaccine, using the RNA virus, yes, you can see the placebo actually has a bad pneumonia, and with the vaccinated monkey, they are protected. And how about the shadow adenovirus carrying spike SARS-CoV-2? And yes, again, the monkey are protected. Of course, the vaccinated one. But if it's vaccinated, the placebo giving a bad pneumonia. Okay, so in a way, it's good. So you can actually protect a vaccinated person from severe diseases. But the question is, can you actually prevent transmission? Can you actually stop infection altogether? So you would imagine when you're vaccinated, you're going to develop tons of antibodies. And when the virus attack, the virus will be blocked by these antibodies. So of course, this is actually the peak, the viral peak. This is actually to describe. So some of the asymptomatic persons, they do have the virus. They start transmitting to other people without symptoms. Now, with the vaccine, should we see this kind of figure? Should we actually stop this viral infection and there will be no more transmission? Is this an idea? But can you see this data? So this is actually the data. This is from the Sinovac, and the monkey has been vaccinated, and then they wait for about a month, and then they challenge with the virus. On day 0, day 3, 4, 5, 6, 7, post-infection. So with the placebo, beautiful virus particle actually isolated from the throat, just like when you do PCR swab. After a week, it's still nicely maintained. But this actually is the situation. This actually, when the monkey is vaccinated with a 3 microgram dose, this with the 6 microgram dose, if you see within 4 days after infection, there is still some significant number of virus particle in the throat swab. So this actually quite disconcerting whether a vaccinated person later on would be protected completely from the infection. Now this is from Moderna. Just look at this region here on the right panel. In the monkey with the PBS given alone, the viral particle, the viral copy is actually shown very nicely in the throat, in the nasal swabs, even after 7 days, it's at control. How about the vaccinated ones? The vaccinated ones, you start seeing some drops, but not completely zero. You get closer to a fairly good, but with a higher dose, 100 microgram of RNA being injected. So again, this has to be very kept in mind that the vaccine, whether they can really actually block the transmission or protect the person from actually transmitting to others. And this from the Shadox one, made by, made from Oxford and AstraZeneca. Again, it's beautiful prevention of the disease, but look at this data. This is actually a no swab even after 7 days after infection. This actually telling the copy numbers of the virus particle. If you see these blue boxes, these are the placebos, and the red ones are the vaccinated ones. If you look at the days, there is no really difference whether the vaccinated animals actually can get protection from the virus in the throat. So again, this should be actually, we have to keep in mind that the vaccine trial, like Dr. Sunil Bahl mentioned, 90% of the case, historically, they fail. So we have to be very careful and make sure that we don't get our guards down. We still have the long haul that the vaccine trial is still going on. So we have to be sure and we have to be careful with our health protocols. So I think that's my last slide. So what are my take-home messages? One, the next-gen vaccine platform have warp speed vaccine candidate. Remember, it's a candidate. It's not actually sure. It's not a certain, but it does actually make things simpler. But of course, we still have to wait for the phase 3 clinical trial. When it comes to protective effects, there are 2 things we're going to measure: whether you're going to protect the vaccinated individual from the disease or the transmission. So we have to wait until this completion of the phase 3. And again, of course, we all want this pandemic to end. But if the pandemic ends in, say, in 3 months, then we probably also going to see the vaccine fail. Why? Because when the trial of the vaccine, you need the pandemic to be ongoing. So I don't mean to be actually an irony, but it's how it works. So of course, that's why what happened in SARS and MERS, the disease is eliminated, and yet the vaccine trial has not even started. But again, at this moment, of course, all of us want to get this end of the pandemic. But lastly, we must remain vigilant and compliant, make sure everybody in the same boat. We have to be compliant to public health protocols. Everyone makes up, physical distancing maintained at all times, and personal hygiene should be done as a personal responsibility. Thank you, Dr. Siswanto, for the time, and hopefully we can address many questions later on. Thank you very much. Jazakallah khair.
Thank you, Pak Ahmad Utomo. I think it is very interesting presentation, especially regarding the new paths on how to develop COVID-19 vaccine apart from live attenuated vaccine like protein subunit and then DNA vaccine, RNA vaccine. And another is the issue questioning to what extent the effectiveness of the vaccine to prevent the transmission of COVID-19. I think this is very important issue because a number of publication also stating that there is cases of reinfection, of the COVID-19 cases of the COVID-19 things. Okay, for the attendee and also all the participants, now we move to the question and answer. Again, if there is a question, just kindly please write in the question and answer chat room, question answer room, not in the chat room. Now we move to a question and answer session. So we have two questions for Ibu Dr. Novilia. The first one is this regarding the phase 3 clinical trial of Sinovac vaccine. The question is regarding the mutation of the COVID-19 that's already presented by Prof. Nyoman. So the issue is, are we sure that our vaccine for Sinovac can cover the mutation, and to what extent is the effectiveness? And the second is from Ibu Setiawaty Balitbangkes. Are there any, for the challenges and the trouble that Ibu Novilia already presented, are there any challenge and problem that unsolved? That's the important, because now in pandemic situation. So please, Ibu Novilia, to answer, to respond.
Okay, thank you, Pak Siswanto, for the first question from Rocky Ban, Pharmacy UTB Lampung, about the mutation. So yeah, I'm really enjoying the presentation from Professor Nyoman. So we have also the same question actually. So when we know about the mutation, the D614G, and then we also try to find out whether this vaccine will be suitable for this new mutation virus. And we find from a journal in the internet that mentioned that even though the mutation is in the spike protein, but not the critical RBD immunogenic part. So not in the RBD or receptor binding dominant part. So for information, to evaluate the immune response from this vaccine, we will do the ELISA assay to prove the IgG to RBD. So this is that we are going to prove the IgG to the receptor binding domain. Besides that, we also have the collaboration with NIHRD, Ibu Vivi and team, to conduct the neutralization assay. So we will challenge the serology from the subject of the clinical trial with the virus that available in NIHRD, whether it's an Indonesian strain. So we can see how this antibody can neutralize the virus. So hopefully this explanation will answer your question. And the second one from Ibu Indah Sulistiowati about the challenge, the unsolved challenging. So I think we can manage so far, but sometimes about the media, sometimes they write something that we are not intend to. So there is a misunderstanding between the media and us. So later we can contact the media and explain that what is wrong about the statement, so they can revise and edit the statement. So it still can be managed even though the media already make a wrong statement. I think that's all, Pak Siswanto.
Thank you, Ibu Dr. Novilia. So now we move to second speaker, Pak Ahmad. I think this very interesting question from Ibu Patrick. Is that right that COVID-19 vaccine already found, and the question is to what extent the effectiveness and the side effect, and then the same question addressed to Dr. Vivi, when the pandemic of corona will end actually, and why the cases is improving so exponential, so large. And then the second is from Ibu Peti, from Badan Litbang actually. I think that's very important question. For how long actually COVID-19 vaccine will protect the person actually. I think that's very important in terms of effectiveness. Otherwise we need re-vaccination, several re-vaccination. Please, Pak Ahmad Utomo.
Yeah, thank you, Dr. Siswanto. That's actually a $1 million question. Is the vaccine actually is found? The answer is, as far as I know, I haven't seen a literature describing that a vaccine has been found. We have seen vaccine candidates. Yes, there are many vaccine candidates, but again, that's why we are still waiting for the clinical trial, phase 3. But again, I think this COVID-19 really teach us a lot about the immunology. Dr. Siswanto also mentioned regarding this reinfection, just recently from Hong Kong, yeah, Dr. Siswanto?
Yes.
Interestingly, actually, with different strain, the vaccine, because it's coming back from Europe, and they got the European strain, the G version, if you will. Again, I mean, this person has actually recovered from the first infection. How come he still get the second one? But again, if you look at what Dr. Iwasaki from Yale, she said that, well, that's actually immunity is doing their job. If you notice that the second infection doesn't give you actually a very difficult or very bad disease. I mean, basically it's without symptoms, so it's manageable. So in the way this is consistent with the monkey studies that when you are actually vaccinated, it protects you from the disease. But actually, the good question would be, will it protect from transmission itself, from the infection itself? Can you actually block the virus from entering the cells in the first place? So that's actually the most difficult one to ask. And hopefully this clinical trial will address that. Because the society will have to choose. Is it okay to be infected as long as you don't get the disease? Or you want to make sure no infection at all? So this actually is very, very interesting, I think, in the next, say, few months, maybe at the end of this year, we should start seeing the result, what kind of data we're going to generate. I don't have the good answer, Dr. Siswanto, but I think we all is going to learn. It's an amazing disease, I would say.
Yeah. Okay, thank you, Pak Ahmad. So I think, I mean, for the pandemic COVID-19, I think now is a good opportunity on how to integrate, between the researcher and then producing knowledge, and then also decision maker also program on how to prevent COVID-19, and then running parallel actually, running parallel. Okay, let me see another question. Maybe there is another question. Okay, I think it's enough. Yeah? There is no more question, but let me see those who raise hand. Oh, sorry. So I haven't seen the other question. Also the attending that raise their his or her hand. Okay, the speaker Pak Ahmad Utomo, also Ibu Dr. Novilia. I think that's a good discussion regarding the real practice on how to conduct clinical trial phase 3 of the COVID-19 vaccine, and then the challenge, also the problem, and then also I think it's very now the committee understand what is clinical trial, and then also they provide voluntary to be the subject. I think that's also the good lesson learned. And then from Pak Ahmad Utomo, there is also another pathway of how to develop vaccine apart from the whole virus, apart from the live attenuated vaccine like protein subunit, and then DNA vaccine, also RNA vaccine, and also how still question actually to what extent the effectiveness of the vaccine.
Oh,
So,
may I add one thing?
Yeah,
if you don't
you
mind?
can add Pak Ahmad Utomo.
Yes,
maybe.
regarding the mutation, that's also in interest of the publics and also the media. There's always a question whether the virus getting more dangerous, which can create a more severe disease. I think in a way, the public should understand that at this point, the virus are actually fairly conserved. The identity is fairly similar. I think Dr. Novilia also has an analysis that RBD sequence mostly are still again conserved. But I think this is very important. Now, the decision of how to make the virus weaken or stronger actually is not depending on the virus, in my mind. It depends on us. If we don't follow health protocols, if we don't maintain distance, if we don't mask up, that is going to make the virus looks more dangerous. So that's actually the key. Yeah. Not so much of the mutation. The mutation is there. The virus seems to be fairly conserved, but it's our own behavior that is going to create a monster, I think. Thank you, Dr.
So yeah, thank you, Pak Ahmad Utomo. So our behavior contribute to the mutation, and then and then change more infectious maybe.
That's right.
Because now the discussion is regarding D614G, isn't it? Okay, all the participants, let me summarize again. Even though Prof. Tjandra already make a conclusion with the previous three speakers, so let me reiterate again. So the first is that it's already happened, the mutation dynamics of SARS-CoV-2 or COVID-19 virus, both in spike protein, but also as well as in the the chain of PCR primer actually, because the chain of PCR primer is very important, because if there is a mutation and then the primer are not already valid, isn't it? Yeah, and that kind of mutation should be taken into consideration for the vaccine development as well as for the primer development. And the second is based on the Indonesian experience. So it is very important to improve the capacity of laboratory network to improve testing. To improve testing is very important in terms of surveillance, in line with the WHO recommendation to better ensure better tracing, also ensure better treatment. And the third, from the Dr. Sunil presentation, So actually, WHO already provide the system on how to accelerate regarding the vaccine development, also to better better provide fairly access of the member state of the WHO member state for the vaccine COVID-19. Also, that's very important. And the issue is to set the priority of those who get priority for the vaccine. And the fourth, now Indonesia is undertaking of clinical trial phase 3 with regard of Sinovac vaccine. Of course, in the hope that that vaccine will be effective and also safe for the people, and then we can accelerate how regarding to end the pandemic of COVID-19. And the last one from Pak Hamad Utomo, I think it's very important for researcher and how to another pathway on how to develop COVID-19 vaccine. Also, it is very important to scrutinize again regarding the COVID-19 vaccine. And we know, I think all researchers know that COVID-19 RNA basically, RNA virus basically is very fast for mutation, like influenza vaccine. Also, that's very very fast in terms of mutation. Okay, thank you very much for the speaker and for the participant. So, to end this session, I would say many thanks and high appreciation for the speaker and all participants for this wonderful international webinar. We hope that from this webinar we have a better insight regarding the progress of of vaccine development of COVID-19. Also the on how to we can accelerate the laboratory capacity to curb the transmission of the COVID-19. And now I will return back the time to the moderator, Ibu Uli. Yeah, please.
Okay. Thank you, Siswanto, for moderating the second session, and also our respected speakers, Ibu Novilia and Pak Ahmad Utomo. Thank you for your important share knowledge and evidences regarding the development of vaccine and the situations of clinical trial of vaccines now in Indonesia. We are reaching the end of this webinar. I would like to remind all the participants regarding the evaluations form. So kindly not to forget to fill it and give your full name and valid email address for certificate purpose. You can see the link of the evaluation form in your screen now, and you can access the link until 6 p.m. Jakarta time. Oh, sorry, 5 p.m. Jakarta time, or 17 hours in Western Indonesian time. And all the slide and your certificate can be downloaded at our website, www.litbang.kemkes.go.id. So you can browse our website and find your certificates and slide your knowledge and your insight regarding the ending pandemic COVID-19 efforts and challenges. And also I would like to thank the audiences for always interested in our webinar. Thank you for your support as well. And please stay tuned to our next webinar, and see you next webinar. Okay, bye.